Thank you for joining us today for the UCD Research Partners Seminar Series. And as many of the regular attendees are already aware, the purpose of the series is to showcase ongoing research in UCD across all discipline areas into SARS-CoV-2, with a view to facilitating greater interdisciplinary collaboration. And today I have the pleasure of introducing Professor Denis Shields, who graduated with a PhD in bioinformatics from Trinity College Dublin, with a focus on molecular evolution. And following his postdoctoral research at the University of Southampton on genetic epidemiology, Denis worked in industry with ICON Clinical Research and with Surgeon Limited. Denis has been in UCD since 2005, where his research has focused on peptides and their proteolytic activation, with applications in food and therapeutics. Denis is currently working on proteolysis modeling and analysis, including sprouting seeds and fermentates, pathogen monitoring in metagenomic datasets, population human disease genome-wide studies involving recombinant detection and involving maternal genetic effects, identification of novel antibiotics, protein motifs and structural disorder, and peptide discovery with regard to SARS-CoV-2. So Denis has a particularly keen interest in interdisciplinary PhD training between computation and biology. And today he's going to talk about his work investigating short motifs and peptides involved in cellular invasion by SARS-CoV-2. So I would like to invite you to submit any of your questions via the Q&A button at the bottom of your screen. And without further ado, I'd like to pass over now to Denis. Thanks.
Thanks very much, Lorraine. So this first slide just shows you that I've been in UCD for too long and I've got involved in too many different things going on. I should really think about shedding some of this by moving jobs somewhere else. But for the moment, these are all the exciting things I'm involved in. So one question that people might have is, why is it we need to increase the number of drugs that could act against coronaviruses? And I guess the answer to that is, we had the outbreak of the first SARS, we had the outbreak of the MERS virus, and both of those were contained epidemics. And then we've had the pandemic with SARS-CoV-2, but in each case, there have been a smallish group of coronavirus experts trying to really build therapeutics that might target some of these viruses. But it's only really when the pandemic hit us that people suddenly said, oh, actually, yeah, that really is quite a good idea. So what are we going to need drugs for? I guess we're going to need drugs if we end up having new strains or species of coronavirus for which there's no vaccines available. And then we might need a variety of drugs rather than just a single all-purpose anti-coronavirus drug, because prophylactic treatment might be needed before infection, or you might need early stage treatment or late stage treatment. And each of those is going to be probably a very different kind of therapeutic, depending on at which stage you think it's going to be appropriate to give that drug or treatment. And then the emergence for many viruses of resistance to treatments means that having a diversity of drugs means we've got sort of a few things to fall back on and try if drug resistant strains emerge. So that's a kind of general perspective on the sort of thinking behind my trying to learn a little bit more about this virus. So I'm not a virologist, I'm a computational biologist. So I've had to read a lot of papers, not all of which I understood in the last year. So one good place for me to start thinking about when I was thinking about this virus was, well, what worked against other viruses? So there's another RNA virus. It's not the same kind of mechanism at all as coronavirus, but it's called HIV. But these kind of mechanisms are what have worked in terms of producing approved drugs that actually work against HIV. So you've got small molecules that can inhibit the replication or the integration of the nucleic acids, targeting polymerases. Then you've also got small drugs that can target the viral protease. So there's a protease in HIV and there's also one in the coronavirus, whose job it is to chop up other viral polyproteins into subproteins. So people have developed small molecule drugs for those proteins as well. And these kind of drugs are great because what these drugs are doing is that they're inhibiting the enzymatic activation, the enzymatic site of these enzymes. So that they can really target very conserved regions of these proteins, which makes them slightly less likely to evolve resistance. And also it's much easier to design small molecule drugs against those kind of pockets within proteins. The other kind of inhibitors are a little bit different, because rather than targeting the key machinery of these the enzyme molecular machines up here. What they're doing instead is they're targeting other processes. Here's a big long drug. You see here, this one here. I've shown you the chemical structure here. In fact, this is a peptide sequence of 36 amino acids. So if you're good at looking at repeating patterns in a structure like this, you realize that there is a repeating pattern of 36 repeats, but then the side bits that come off are the amino acid side chains, which are kind of variable depending on which of the 20 amino acids are at a particular position along this sequence. So this is a very interesting, I'll come back to this class of peptide later, but this is a peptide which is involved in interacting with parts of a large viral protein that's involved in the fusion of the HIV virus. Then there's drugs that block the host cell receptors. So the virus sticks onto the cell, and then the cell has these receptors on it, and there's an antibody-based drug against CD4, and then there's interestingly a small molecule drug against a chemokine receptor called CCR5. So these kind of drugs small molecule drugs are much more attractive because they're easier to get into the body of a patient and have less stability issues, whereas a big long drug like this one here needs to be injected, as would an antibody like this would need to be injected. They're not going to cross the cell membrane easily. And then finally, there's another class of drugs which block the viral surface protein. So all of these drugs aren't simply investigational. These are drugs that have actually entered the clinic for HIV. And HIV, as you know, had a really poor track record in terms of developing vaccines against it, which is why so much effort went into these drugs. And the best therapeutic strategies for HIV then are combinations of these different drugs. So I think from this, this kind of gave me some perspective on where to start thinking about where to go in terms of drugs against SARS. So my particular area of interest is in peptides, because that's where I've been doing a lot of research. So I'm going to focus more on that. So here's a picture of the virus. You might recognize this here. This is a spike protein sticking out. These little liney bits here, that's the phospholipid bilayer, the membrane that surrounds the virus. And we have a very similar phospholipid membrane surrounding our own cells. And the fusion of the viral phospholipid membrane with the host phospholipid membrane is one of the key events that's involved in the entry of the virus into the cell. And this big spike protein has a key role to play in this. And there's various other proteins in the actual virus. And then in addition to those proteins in the virus, there are also a number of proteins which aren't in the viral particle, but that the virus then makes when it gets inside the cell. See, this is a kind of linear view of the viral genome, and these are all the proteins that are encoded by it. It's a big protein here that's later chopped up by a protease, you might remember me saying. So this can be chopped up into lots of subproteins, and this is the kind of pattern. This S1 here is that spike protein I mentioned to you earlier. So you could consider all of these proteins as potential targets for viral therapies, some of them better than others, some of them more strongly conserved than others in evolution, and therefore slightly less likely to evolve a rapid resistance. And that's sort of what we were wondering about when we were looking at this. So this is actually for the original SARS genome. SARS-CoV-2 is very similar. It has some differences around here, but largely a very similar genome organization. So a way of thinking about how to inhibit the interactions, you might remember I was saying that a number of the drugs work by inhibiting the interactions between proteins. So if you block the cell receptor, you might stop the virus binding. Or if you block the viral particle itself, something attaching to the viral particle itself, that'll stop it binding to the cell. Or alternatively, you might block components within the cell that interact with each other. So it's a good question. What viral proteins, which of them interact, like the SARS-CoV-2 proteins, what do they interact with? What other SARS-CoV-2 proteins do they interact with? Could we inhibit their interactions and mess up the virus communicating with itself in some way? Or could we target the interactions with the human proteins? So this is just a sort of simple schematic of, there's a spike protein. I think this is meant to be the membrane here. Yeah, and here's a bunch of different proteins that are in the membrane of SARS-CoV-2. And then you might imagine there's various host proteins that come in. So a lot of these, interestingly, just bind to short motifs like the TMPRSS2 and furin are both proteases of the host that seem to have potential roles in activating the virus. ACE2 is the most important known receptor. There are other potential receptors. I'm going to talk a bit about integrins and about, I won't talk about today, but neuropilins, another very interesting potential receptor. And then there are all the viral interactions among themselves as well. And there is a database called Intact where you can get a listing of all the virus interactions involving coronaviruses, and there's over 7,000 of those. So you could consider there's quite a lot of potential targets to go after, and which ones should we focus on? So traditionally, people would have said targeting protein interactions is a waste of time. Smaller drugs are really desirable, and it's quite hard to make a small drug to target a protein interaction, which might be over quite a large surface. So from this point of view, short motifs in proteins that are involved in interactions might be easier to mimic with smaller drugs. Now we know, and from our collaborators of mine have done studies where they reckon that out of the 20,000 human proteins, there might be 1 million of these kind of short little linear motifs, mainly involved in protein-protein interactions of one kind or another within them. So these would be short regions of the proteins that play critical roles in the contacts between the proteins. So a classic example of these, and this is the one that really got me interested in this area, I was working years ago on a drug trial of drugs which were antiplatelet drugs, and they were chemical compounds which were modeled on this arginine glycine aspartic acid, a 3 amino acid short linear motif. So that's really what I mean by linear. It's just a little subsequence, and short means 2 to 8 amino acids long, thereabouts. And this particular one, it's an exact, it's got to be R, G, and D there. There's no variability. A lot of other motifs might have quite a bit of ambiguity in their positions. So this particular sequence is found within the larger protein fibrinogen, which is very important in clotting. And we know that this binds to integrins. And people have developed drugs. So this is a cyclic drug where they took the linear sequence and they made it much more stable by cyclizing it. But if you've got a keen eye, you'll see that that looks like an acidic group. That's aspartic acid. And this is the arginine group here. And the glycine group is there's nothing at all coming off there because glycine is really just a hydrogen. So you can't see it there. But this essentially is a chemical compound derived from a short linear motif. So can we tackle SARS-CoV-2 with slim-based drugs? And I think the answer is there's lots of potential. And this is from a review that Norman, he did a PhD in the Conway years ago, and he also came back and did a Surge fellowship here, and he's now in the Imperial Cancer Research Centre in London. But he wrote a really nice review on short motifs a few years ago in viruses. And I'm not going to go into this slide in any detail except to say every possible process where you think viruses might be doing something at all, entering, escaping, signaling, degrading proteins, controlling immune response, cleavage, transport, cell cycle, all these things, viruses are using motifs to alter them. So we know viruses are using this very effectively. So can we turn the tables on the viruses and actually try and identify motifs that we think are important to understand better the viral biology? So this is a rather simple example that I chose because this is a short protein from the virus called the ENV protein. I chose it because it's a bit easier to look at. Here on top is that sequence in a 1-letter amino acid code. And that is for the top line is from SARS-CoV-2. And below it are 4 other pathogens, the original SARS, MERS, and HKU1, which is a common cold virus, I think. Okay, so this is an example of a protein where you can see it's on the outside of the virus. There's a bit that spans the membrane, and then there's a bit that is on the inside. And then you can put it into this software from my collaborator, Toby Gibson, has developed, and it'll tell you where all the motifs are. The only thing is that a lot of these motifs are false positives because there are many, many motifs appearing in really quite a short sequence. Some of them you can exclude easily enough. You'd say, oh, it's very unlikely that the intracellular part of the protein would have a glycosylation motif. So I can eliminate those 2 quickly from inquiries by allowing for context like that. And so we applied this software in collaboration with Toby, who runs this website, and we made some predictions. And we wrote a paper about it, which appeared recently last January. So I just sort of came on board this study kind of a little bit late on and joined in because I had some questions of my own, and Toby thought it'd be good to actually get involved in the same paper. So what we did here, and this is really just a summary of what we found, was we just tried to look at, here's the virus up here, and that's its phospholipid membrane there. And on the outside of it is the spike protein. It's shown here as a trimer because the spike consists of 3 copies of the same protein. And on the surface of this, we found an RGD motif, which I think I mentioned earlier, but lots of other motifs involved in binding to neuropilin, which we know has a role in the infection of SARS-CoV-2. If you inhibit neuropilin, you inhibit entry, and various other interesting motifs. And the focus of this study kind of shifted a little bit more towards looking at the intracellular tails of the receptors, so the integrin and the ACE2 receptors. So the ACE2 receptor, that tail had not really been studied a whole lot, the biology of this tail, because it's very much viewed as ACE2 is a receptor, virus binds, it just gets in, or ACE2 has important roles in biology, nothing to do with the virus, where it's a protease that chops up other proteins outside the cell, but that's not really important to its viral entry as far as we can tell. So what our study found was these interesting motifs on the inside here. So there's quite a lot of detail here on what all these motifs are. I'd like to just focus on this Lear motif, which is involved in a process called autophagy, which we see in both the ACE2 here and the integrin tail. So, and these processes to do with integrins and actin regulation and autophagy, a lot of them are to do with this question of how the virus is endocytosed into the cell after binding to the receptor. So this receptor here, ACE2 and integrin, they would signal for their own internalization upon signaling, and they'd be re-trafficked around. So the question is then, is the virus essentially using some of this machinery on the inside of the cell to help get here? Now, autophagy might sound like something you'd think you didn't want if you're a virus, because autophagy involves the digestion of parts of the inners of the cell, and is often used by the host to try and restrict viruses. But in fact, the coronavirus uses autophagy as a place to hide its double-stranded genome by going into these autophagy type vesicles, which seem to help it evade the host innate immune system, the sort of natural mechanisms it has to attack double-stranded RNA. So it's quite hard when we look at this autophagy to think about, do we want to increase it or decrease it, because we don't quite know how that's going to impact on the viruses. So here we are looking in a little bit more detail then at those motifs I mentioned. I mentioned this RGD motif, which we know has the potential to bind integrin. And what I think was very intriguing was when we lined it up with related viruses, we found in one of the common cold viruses from human, we found an NGR motif. And you say, well, that doesn't sound very similar to RGD. But the reason this is interesting is that this binds to exactly the same site as RGD once that N, that asparagine, is chemically modified to isoaspartic acid, which is a natural degradation process that happens when you've got an asparagine followed by a glycine. So we believe this region has interesting potential, that it might have evolved twice to be a receptor for integrins. The difficulty I think we have at the moment in going much further is that people have been looking at this experimentally, but there's only really rather preliminary evidence that this RGD binding is important in integrin interactions of spike. And indeed, one of the inhibitors people have found against the spike for integrin binding has actually not been the RGD region, but another region of the integrin. So I think the jury is still out on how important this is for the critical entry of the virus. It may have some other role in the later pathology, so we've got to also bear that in mind, because RGD interactions with integrins in later cell types may be much more important than the initial cell entry into the nasopharyngeal or lung cells. And here is a similar picture. I'm not going to dwell on this, but this is for the ACE2. And with human proteins, you can look at evolutionary conservation to give you a clue about whether the motif is true or false. It's much harder with viruses because they evolve functionality, so they gain and lose them much more rapidly than humans. But for humans, you see this motif conserved here over evolution, you say, aha, this is very likely to be important. And that's what we believed, and we were lucky because collaborators, Ilva is a good friend of Toby's, and she then validated some of our findings in the laboratory and published a paper alongside our paper, which indeed validated that some of these motifs do bind to host proteins. So some proteins have their domains which have evolved to bind specifically to those motifs. So she was able to choose the sets of domains to test and then discovered which were good. So just to sort of wind up on this, where do we go from here? We've made some interesting findings. They're making us think a little bit about why the virus is using those receptors. Why is it using ACE2 and integrin? Is it possibly, and possibly integrin? Is it because the internalization motifs of those receptors is actually aiding them in getting to the next place within the cell where they can do their dastardly deeds and replicate safely without challenge? So currently we have a project, Mark Devosel in the College of Surgeons and Virginie here in UCD, along with Ajay and Niladri, are working on this project. And really what we're trying to do is we're trying to go from a computational library of peptide designs to really see, can we find some short peptides that might mimic these linear motif regions, and then test them in the lab to see, are they going to inhibit SARS-CoV-2? That's in a fairly simple cellular assay. There are some limitations to that because ultimately what these motifs may be doing in a simpler cell assay may be different to what happens in vivo. But that's our screening process. And just to wind up on this, have a little think here about some of these images of what's going on. We've got a virus here, which could be entering a bilayer. So this is a molecular modeling of what might be happening with some of the proteins involved in flu, which you might think might have a similar effect. But what happens here is that parts of the protein, like the equivalent of their spike protein, are embedded within the virus, but also other parts then embed within the host protein. And somehow these central parts kind of move away, and this whole process seems to manage to slurp together the virus and the host protein. So this is how the fusion seems to occur when you're looking in a model system, and may well be what kind of thing happens in real life. It's always a little bit hard to tell because these proteins are enormously complicated. So this is a kind of a molecular simulation for the SARS-CoV-2 protein. When you see the blue balls kind of extending out towards the right, that is when it's in its pre-fusion state. But then when it's in its post-fusion state, after its integration to the membrane and that kind of conformation, you can see there's a massive conformational change undergone by this protein. And the question is, how do we find drugs that can target that? And you might remember the HIV drug that I mentioned earlier, the fuseon and fervitide protein. So what that does, it targets, you see this yellow bit on the outside here, it takes a stretch of that, and then that probably binds to these, the orange bits in here, which are those alpha helices there. And it seems that those bundles of alpha helices form to somehow be a critical part of this molecular machine that's undergoing the transformation. And by making a piece of this peptide here, it seems to bind to this part and then prevents the correct formation of the post-fusion state. So people have tried a very similar thing in SARS-CoV-2 because, very curiously, even though HIV is a retrovirus with a very different mechanism of transmitting its genetic information, it shares many features with the SARS-CoV-2 in terms of the structure and organization of that entry protein. So the HIV protein, which had that drug designed from it, is similar in many features to the spike protein on SARS-CoV-2. And so by following on what happened with the HIV work, they took the region from spike. So there's a sequence, an amino acid sequence. They synthesized that as a synthetic peptide. They tried various kind of modifications, the most important being it's adding this cholesterol lipid. And then they tested one of these in an animal model, in a ferret model, where they infused it intranasally. And this really seemed to prevent the spread of SARS-CoV-2 in an animal model. So there's some very nice things about this. It validates that peptide delivery is possible in a not systemically, but actually in some kind of intranasal or possibly inhaled infusion. It also shows that what worked in HIV does also work for SARS-CoV-2. However, there are some problems with this going to the clinic. I guess maybe its potency may not be quite strong enough. The other thing is it's quite big and expensive to make. And also these large peptides are probably more likely to evolve resistance mechanisms within the spike protein because they have so many points of interaction. If you remember from the previous picture, there's all this region here, this long orange region here could all maybe evolve slightly differently to make it interact less efficiently with the therapeutic. So it's given us a lot of inspiration that this work has recently come out to push away with what we're doing. What we're really looking at is trying to make much smaller peptide-based drugs, which ultimately might be a little bit more like small molecule drugs, and also then look at how we, protect and deliver those smaller compounds in the cell. So that's it, and I'd just like to thank the people who are currently working with me on that SARS-CoV-2 project. So Virginie Gauthier here in UCD has been critical because she has the screening facility for testing whether any of the peptides that we synthesize actually can inhibit the growth of the virus in cell lines. Mark Devasel is a peptide chemist who I've worked with a lot on antiplatelet peptides in the past. And so it's really excellent to be back working with him on this project, along with Neil Adry, who works in Mark's group, and Ajay, who's working with me here in UCD. I'd just like to thank all the people who, Balance and Toby especially, who put so much work, and Lucia, who put so much work into that SARS-CoV-2 paper I described, and also my former group members, Norman and Rich, who put so much work into the software I use every day. Thanks very much.
Thanks very much, Dennis, for a very clear presentation with some very nice images. I have a couple of questions here from Sean Lestrange. So is HIV a good analogue for SARS-CoV-2, given the fact that HIV is a retrovirus with no proofreading, whereas SARS-CoV-2 is an RNA virus with proofreading?
Yeah, I mean, I think that the big thing, if you're referring to proofreading, the big thing is obviously about the evolution of resistance. And we don't really know a lot about the evolution of resistance to coronavirus drugs because there haven't been so many coronavirus drugs out there. We do know a little bit about sort of evasion. Potentially, we know a little bit about evasion from the natural immune, the specific immune response, in the sense that there might be some suggestion that would say, like what's happening in Brazil in Manaus in terms of the evolution of new variants, there may be variants which are good at infecting people who've previously been infected. So we could see that the virus, I think the virus is not going to evolve as rapidly as HIV, so it's not as terrifying in terms of vaccine production and not as terrifying in terms of drug resistance, the rapidity of drugs resistance. But I still think we still have a similar issue there. Regarding whether it's a good analogy, I think for some of the example that I gave there of the long peptide, which was pretty much designed based on the HIV one, shows that in that particular case, yes, you can. But there are many other aspects that clearly you can't. For example, HIV is a retrovirus. It's got an integrase. It's got various bits of machinery that you don't have in a coronavirus. And there are other things in coronavirus that we could possibly target that aren't in HIV. So it's more really for just thinking about feasibility and practicality. And they are both, they have some similarities. And I think that whole fusion mechanism is surprisingly similar given how diverged the viruses are. So it's certainly good to look for inspiration, I think, from other viruses like HIV, but they wouldn't be good models for the compounds.
Fair enough. And I suppose Sean is puzzled about the fact that SARS-CoV-2 is single-stranded RNA, not double-stranded.
Yeah, I mean, I don't quite know why all the different viruses in the world are different to each other. And there must have been enormous selection constraints if they've evolved from each other. Like if it's a little bit hard to tell, but I don't really understand. I don't know much about the evolutionary origins of these things, but if, say, they evolved from each other and then they've evolved totally different mechanisms of passing on their nucleic acid information, there must be incredible evolutionary constraints on to make them do that. And I guess that might be the host response in terms of host acting against double-stranded RNA, which it detects as a bad thing. So I could see the motivation to have an infectious particle with a single-stranded RNA, because when the single-stranded RNA appears in the host cell, it's less likely to immediately be flagged as a clear enemy compared to, say, a double-stranded RNA. But exactly why particular families are single and double-stranded, I don't know.
Yeah, it's interesting. And you talked about a number of potential mechanisms that the virus can obviously exploit to enter the cell. Does this mean that you would need more than one inhibitor being used at the same time to be effective?
Yeah, I think that's definitely a possibility. It's a little bit hard to know until you try things and see what happens. I mean, I think one of the reasons I think the HIV went for the multiple factors was because of the problem of resistance, which is a combination of two factors: the rate of evolution of HIV, and then the fact that HIV is a chronic disorder where you have it for a very long time. So having the combinations makes it much less likely for the resistance to sort of emerge. One thing we're trying to think about is whether we can sort of combine some activities within a short peptide, but it's not clear how feasible that is.
Okay, and I suppose one just final question, and I suppose to a certain extent you've answered this maybe, but it's around, you know, the fact that SARS-CoV-2 is obviously contagious 48 to 72 hours before clinical symptoms appear. And I suppose, you know, 44% of transmissions are actually occurring in that pre-symptomatic phase. So, you know, when you were talking about the ferrets, I suppose you were talking there about some sort of a nasal mechanism for using them as prophylactics. So I assume predominantly they will be used as prophylactics. Is that fair to say?
Yeah, I'm guessing when you start to think about those drugs, you'd imagine that if it's quite hard to time things, so prophylactics makes more sense, so that then you'd have. But yeah, I think the economics of all of this probably needs to be worked out yet, because it depends very much on if you have prophylactics which are a relatively cheap and sort of effective drug without too many side effects. You know, they might be easier to sort of develop and put into clinical practice. But if you've got very expensive ones that are difficult to synthesize and then, you know, give for a long period, and then you've got to look at their safety profile as well. So I think there would be a lot of things that would need to be worked out before you could translate some of what we're doing in terms of research into something in clinical practice.
And I have one final question here, actually, from Sean. He's asking how costly would these drugs be, and how many administrators would be needed. For example, administrations rather would be needed, a dose a day, or that's.
I think that
week,
the enfervatide, the Fusion HIV drug, I mean, I think one of the main reasons it sort of fell from use was that the patients just hated the sort of daily injection of the drug, and because it wasn't an oral drug for HIV. Now, with a respiratory drug, and if you're giving it for prophylactic reasons, certainly you probably wouldn't be looking at systemic delivery. You're probably looking at inhalation or a nasal infusion of some kind. And how often you'd need to do it, I'm not quite clear on that. I don't know, actually.
Okay, Dennis. Well, I think the fact that we're running over time, we'll close the session there. So thank you once again for a very clear and stimulating presentation. Very enjoyable. And just to say to everyone, this is the last seminar in our current series, but we will keep you posted regarding any future presentations. So many thanks again for joining us. Thanks, Dennis.
Thanks everyone. Bye-bye.
Thanks.